brain. They are the backbone for, how do I move to the next slide? Oh, this one. Oh, this. Yeah. They're the backbone for many fundamental processes like how we perceive things, how we think, and also this dynamical property can get altered in various neurological diseases like chronic pain, Alzheimer's disease, epilepsy, and so on. And if we can understand this dynamical behavior better, we can also develop better treatments for various neurological diseases. Some treatments which can really improve if we understand these dynamics better. One of them is called a deep brain stimulation, where we essentially stimulate a part of the brain. And we can also develop better neurotechnologies. For example, the brain-computer interface, where the whole idea is that you don't really do anything, but you think in a way to maneuver something. For example, a robotic arm in this case. So overall, there are broad applications. From a fundamental point of view, we just want to understand better on how we think and perceive things. But also, if we understand these things better, we will also be able to understand diseases and develop better treatments for them. So those are all the broad applications, and that's why it's important to study the brain dynamics, how the brain functions in general better. Now, there are various ways of studying it. There are various neuroimaging technologies available that can really capture how the brain signals look like in different conditions. So that's one way of studying. But whenever they are dynamic, there is also a lot of mathematical modeling that you can do to try and understand how dynamical behavior evolves under different conditions. So most of my work that I will present to you is going to be based on mathematical modeling of this kind of a dynamical behavior. Now the main thing we try to address with this kind of a mathematical model is a fundamental question in neuroscience. And that is that the brain structural wiring is quite static in the sense that it doesn't change over a range of years. But the functional activity is really dynamic and it changes over a millisecond level. Like how my brain function looks like is different when I am talking right now versus when I am sleeping versus when I am meditating or doing nothing and so on and so forth. So what's the relationship between the static constraint, which is the structure, and this dynamic brain function? That is something we try to study with mathematical modeling. So I'll start with a brief primer on what structure is and what function is, and then I'll go into some mathematical modeling we've done. So structure is something we try to evaluate using a technology called diffusion MRI. It's based on MRI, magnetic resonance imaging, and it tries to identify how the anatomical tracts look like. In this sense, what are the wirings that are going from one region to another, in what direction and to what extent. So that is something we can get from an MRI based technology, and once we have this wiring information, we can project it onto a template. The template basically divides the brain into a fixed number of regions with fixed boundaries. So once we project this wiring onto the template, we get a network. So this you can assume is like a graph where different nodes are regions of the brain and they are connected to different nodes, and we know the extent to which they are connected based on the number of tracks that are going from one region to another. So we get this network, and finally there are two matrices we get. One is called a structural connectivity matrix. Every region of the matrix is the extent to which two different brain regions are connected to each other, and we get a distance matrix. Every region of this matrix tells us the distance between two different brain regions. So these two matrices is what encompasses structure for us, and in all the work that I am going to present, so on and so forth, by structure I really mean these two things. It's volumetric. Yeah. Yes. Yes. What about brain function? Now brain function, there are various different neuroimaging technologies available to capture brain function. The popular ones are MEG, EEG, magnetoencephalography, electroencephalography, and functional MRI. All of them try to capture the electrical activity of the brain. Functional MRI indirectly and MEG and EEG more directly. And the idea is that we project it onto a template and we get some source reconstructed time series. So these are just time series corresponding to different brain regions, and that's brain function for us. From this we can extract various features that might be of interest and compare compare between two different conditions. Now the whole idea is, given the structural network, can we write down a mathematical model in terms of some model parameters?
If there are redundancies, let's say in terms of loops and stuff like that, how is that encoded using the structure matrix, like the adjacency matrix and the distance? Because let's say there's more than one trajectory connecting 2 points.
Yeah, so in this, no, it's not that fine-grained. It's really telling you total what are the number of connections between two different regions. So we don't incorporate that.
Adjacency matrix is based on experimental data.
Yes, this is also based on MRI. Both the matrices come from experimental data, real data.
We know that the distance is proportional to the physical distance. It could be going in a tortuous path, right? So I'm saying that the distance between 2 points is necessarily a linear line joining the two. It can be quite tortuous.
It can be.
So how are you taking account of that?
So the way this technology is based on tracking diffusion of water molecules along a path. So if it's tortuous, you can capture that. Alright. So the whole idea is, given the structural network, can we write down a mathematical model in terms of some model parameters that can not just accurately capture brain function, but also predict how brain function can change under different situations. So one of the models that I worked on when I was a postdoc was called a spectral graph model, and that tries to do exactly this. Given the structural network, write down a model and try to capture brain function and then see how the brain function changes under different conditions. Now, the main aspect of function that we try to capture is actually from the time series. We look at the frequency components because that's of more interest for the applications we were interested in. So by function here, so on and so forth, we are going to look at the frequency, everything in the frequency domain. So I'll start with explaining what this model looks like. I'll give you a brief overview and then we'll go into the math and some applications. So this model is a hierarchical model. It consists of two levels. So at a local mesoscopic level, which is a lower level, we model two kinds of neuronal populations in a brain. One is called excitatory and one is called inhibitory. So as the name suggests, excitatory really it means that it increases the overall signal, and inhibitory inhibits that signal. And there is generally a balance between the two, and they interact among each other. So that's what we model. And that is something we model for every brain region. And then there is a macroscopic scale. At the macroscopic scale, we model the long range neuronal connections. These are really long neurons, and these are the ones that really connect different regions. And these are primarily excitatory neurons. So this is the entire structure of the model, and we know the structural information is taken into account at the macroscopic level because we know the extent to which they are connected and the distance between them. So this is the structure of the model. When we simulate it, we directly get the frequency information that we are interested in. We basically get what the power spectral density looks like for every brain region separately. So every line here corresponds to the power spectral density of one particular brain region. So the number of lines really depend on how many regions you have divided your brain into. And every line tells you the frequency component, the frequency richness of that particular brain region. So that's what we get from the model, and we want to compare it to data. What's a real data like? In this case, and today all the examples I'll show you, the real data will come from MEG, which is called magnetoencephalography, which is just one way of capturing magnetic signals of the brain. We do some pre-processing, frequency transform of the time series, and finally we get how the power spectral density looks like for the real data. Now the idea is, given the real data, how do we make sure our model is as close to the real data as possible? Because that's when it's capturing what we see in the real data, right? To do that, we can do an optimization and find out what are the optimal model parameters such that the model output is as close to data as possible. So that's what we do, and here the advantage of this particular model really shows up. there are very few model parameters that we need to really optimize for, and they are all global in the sense that they are the same for every brain region, and they all have some meaning. So at the mesoscopic scale, you have some excitatory and inhibitory time constants, you have some gains, and then at the macroscopic scale, you have a coupling constant which regulates the extent to which different brain regions are coupled to each other. Then you have a parameter for speed which regulates the speed at which signals are transmitting from one brain region to another, and then you have the long range excitatory time constants. So these are all the parameters. This is all we need to optimize for, and even with just such few parameters, we are able to get a good match with the data. So let's go a little bit into the math. I'll show you how the equations look like before actually showing you the model, how the model works. Yes.
So what is the meaning of this frequency versus power graph? So for example, brain can give a signal, we can have thoughts, or brain can give a signal to the body part to do something. So how does that thought is captured in this power versus frequency thing?
So this is actually just capturing whenever we do some neuroimaging recording, it gives us time series. So all we are doing is Fourier transform of the time series, and from that what does Fourier transform do? It divides the signal into sine and cosine curves and tells you which frequency is most dominant in that time series, and that's the information we are getting from here. So finally what we do is we compare this under different conditions. For example, in different cognitive processes, when somebody is focused versus when somebody is not doing anything, how do the frequency components look different? Does that tell us anything?
Okay, so these are the only two parameters that we are trying to map whatever is happening in the brain into this region.
Yes, for this whatever applications I'll show you in this.
And
yes.
one more question, more philosophical. So you're trying to build a model for brain. Does that mean that it's not it's like predetermined for a given stimulus, it's going to behave in a particular manner, we don't have any control over it?
So that's not necessary. Firstly, there's a lot of inter-individual variability, and
yeah, for an individual person, let's say for one person you build a model, does that mean it's deterministic? You give a stimulus and we are going to expect this particular signal from the brain?
it depends on whether a particular region is controllable or not. I think Ramakrishna is sitting here who can answer this better. But not every, for example, there are certain brain states, not every brain state will be realizable, and not every node can be controlled in same way as other nodes. So it's not a given. Depends probably on the controllability parameters, if I'm correct.
We can discuss later more on over coffee.
Yes. You had a question.
This graph suggests that different parts of the brain they respond similarly to the same excitation. Like the graphs, you said the lines correspond to different nodes, right?
Yes.
In the graph, so they all look pretty much the same. So does that suggest that all the regions in the brain respond similarly?
So that's not the case. This model is certainly a simplification. If you look to the right, that's real data, and you see there is some heterogeneity, and you see a lot of heterogeneity in the power, right? So they don't have the same power.
Okay, so that response depends on the how much intense that is.
Yes. There's another question at the back.
Sorry, I just had a quick question regarding this optimization workflow that you. So the model that you're trying to fit is non-linear, right?
It's actually a linear model.
Oh, I see. Okay.
Yeah, I'll show you the equations in the.
So just for me to confirm, so if it's a linear model, the number of constraints and the number of free variables that you have do match. In the sense, there's no non-uniqueness associated with the optimization itself.
No. Okay, so I'll go ahead. In fact, I'm going to show you equations in the next slide. So this is how the differential equation looks like. Let's say X is the macroscopic signal of the kth brain region. So it essentially consists of three terms. So first you can say it's like a self-decay term we are convolving with some function that captures the macroscopic level experimental variability. Second term corresponds to signals coming from other brain regions that are connected via the structural connectivity matrix, and they are all coming with a delay depending on the distance between them and the speed at which they are transmitting. and they are proportional to the coupling constant, which regulates overall how much we want to scale the incoming signals. And third one corresponds to the mesoscopic or the regional level excitatory and inhibitory signals. So the nice thing is you can get a closed form solution of this in the frequency domain, and you get an analytical solution. All you have to do is take Fourier transform of this. This is how it looks like in the vectorized form. So now it just becomes a linear algebraic equation. All you have to do is apply some linear algebra and solve and rearrange the terms, and then just solve for x of omega, which gives you what the signal looks like in the frequency domain. So that's all we do, and when we do some rearranging, we come across this term called, we call it a complex Laplacian matrix, but it's essentially similar to a Laplacian matrix you might have seen in graph theory, and we do an eigen decomposition of that, and this is how the equation, final analytical solution looks like. So we don't need to understand this analytical solution, but it's just to keep in mind that with this set of equations, we can directly get x in the frequency domain, and we can directly compare it to the data. We don't have to do time consuming simulations in this case. So, computationally it's quite fast and very feasible to do. And these are the eigenvectors and eigenvalues of the Laplacian matrix. So keeping that in mind, but does it do its job? Does it actually capture real data? So first I am going to show you just its performance in case of healthy individuals. They are all in resting state, in the sense they are just sitting with eyes closed. They are not sleeping but also not actively doing anything. That's called the resting state. So in black is MEG and in blue is what we get from the model. So you can see that it matches the shape quite well. It also to some extent matches the spatial patterns, but there is room for improvement. So that's one of the things to work on. We have also done some applications of this. This is like a linear equation, so we looked at the stability properties of it in the Laplace domain, do Laplace transform, look at poles and complexes, poles of the system might be similar to people who are coming from chemie background. So that's to get a theoretical understanding of the model. Okay, so what I've shown you here till now, this model fits well to healthy MEG data. We can understand to some extent its theoretical properties. What's the use of this kind of a model? So next I am going to show you an application to show how it can help in trying to understand the underlying properties of the brain. So the application today I will show you is Alzheimer's disease. So Alzheimer's disease, you might know, is a neurodegenerative disease characterized by cognitive impairment, memory loss, among other characteristics. And one of the things about Alzheimer's disease is if you do a neuroimaging recording of a patient with Alzheimer's disease, you will clearly see there is some abnormality. But what are the underlying mechanisms that might lead to that abnormality? That is hard to infer just by looking at neuroimaging data alone. That's where using any kind of a mathematical, little biophysically realistic mathematical modeling can help. Now, finding these relationships between mechanism and what you see in recording, that is something you can do only in basic science studies with for example, a mouse rodent model, but not in humans where you can't do invasive studies. So that's really the knowledge gap, and that is something we try to bridge here with this model, and I'll show you how. So let's look at real data first. If you look at the MEG recordings, in blue is healthy age matched controls, in orange is patients with Alzheimer's disease. So you can clearly see, right, there is a difference between how the spectra looks like for patients with Alzheimer's disease versus healthy controls. So yes, if you do an MEG recording, you will see there is a difference. But why does this difference arise? That is something we will try and answer by applying the model that I showed you to this data, and we will see how the model parameters look like, and we will compare them between health and disease and see if that tells us anything. So this is the data, and on this we will apply this workflow, and we will try to find out what these model parameters are like, and we compare them. So when we compare, we see that some are statistically quite different, and there are two time constants. One is tau g, which is the long range excitatory time constant, and another mesoscopic excitatory time constant. They are much longer in Alzheimer's disease, and then this inhibitory gain is also higher in Alzheimer's disease. overall, what these parameters indicate is that there is some abnormality, impairment corresponding to these particular parameters in Alzheimer's disease that might result in the difference in MEG recordings that we see. Most stark difference is in this particular parameter tau g, which is much longer in Alzheimer's disease. I want to highlight this because its importance will show up later, but just to remember, this one is it's quite different between the two populations. Okay, so this tells us, there's most likely, there is some impairment in the long range excitatory neuronal time scale. That's what tau g corresponds to, and that might be there in Alzheimer's disease. But do these parameters have any significance in the sense that can they be used as markers of disease? To test for that, we developed a machine learning classifier with all these parameters as features of a classifier, and then we tested if we can use these parameters to classify Alzheimer's disease from healthy controls accurately. So this is how the ROC curve looks like for classification, and the area under the curve you can see is 0.85. So it's quite accurate in classifying Alzheimer's disease from controls. Not just that, again tau g, the parameter which was most different, is the most important parameter for classifying Alzheimer's disease from controls. All in all, what this indicates is that you can use these parameters also as markers of disease. They can accurately classify Alzheimer's disease from healthy controls. But do they have any clinical significance as well? So the next thing we wanted to test was for that, by testing for if there is any association between these parameters and the cognitive decline in patients with Alzheimer's disease. So we tested for the association of these parameters with global cognitive decline scores in patients with Alzheimer's disease, and interestingly we saw unique association. Again, it's just the tau g which stands out, which is uniquely associated with the global cognitive decline scores in patients with Alzheimer's disease. This particular score is called a mini mental state exam score. It's just a score which tells you the global cognitive decline. So this score actually, when it reduces, it indicates more cognitive decline. So what this plot is indicating is that there is an increase in tau g, and that's associated with more cognitive decline. Or in what it's trying to tell you is that there is an impairment in the long range excitatory neuronal population, and that is associated with global cognitive decline. Now this is quite intriguing, because human neuropathological studies indicate that the pathology that impacts global cognitive decline preferentially affects excitatory neurons, and we see this unique association even here just using raw neuroimaging data and doing a modeling on that.
So Parul, sorry for not understanding this figure, because as chemical engineers we are often used to much more deterministic data, because the data does look a little bit, much more scattered than this. So can you try to explain, how can you say that this clearly indicates
Yeah. So yes, that's a great point, and a lot of people ask this. With human data, a lot of times we can't get something better than this. First, the challenge is how many number of subjects can you recruit for a study. Here we had 88 patients with Alzheimer's disease, 88 healthy controls. I guess if you increase the number of subjects, you'll see less scatter. That's one thing, and the other thing is, because in all of these human studies there is so much variability, we look at the p-value to see if it's significantly, if the slope is significantly different from 0 or not, and that's why I've indicated the p-value here, because it's significant. We say that there is an association. That's all we care about, whether there is a non-zero association or not. A p-value is, to go back to my statistics, so when we do hypothesis testing, if a p-value is less than 0.05, it indicates that you can, in this case, the null hypothesis is the slope of this graph is 0. So when p-value is less than a threshold, you can reject that null hypothesis, and so we can reject the null hypothesis that the slope is 0, and that's why we can say with some confidence that it has a non-zero slope.
What is the number of samples? There are only 88. So I don't know whether the threshold of 0.05 is for 88.
So
we have 8,000 or 88,000, maybe that's more statistical.
So, yeah, well, it depends. But p-value 0.05 is a very standard threshold used to.
So for large number of data, right? You have the aggregation, and then if you're talking about persons, there can be persons with certain variability, right? So
Yes.
all that, to claim anything is difficult.
So, I mean, this is an adjusted p-value. After adjusting for multiple testing, that too we used a quite strict adjusting conservative approach, which was the Bonferroni correction. So, given that we use something quite conservative and still it turns out to be significant, that's why we were fine with this. And the scatter of the points, I mean, we can't do much with human data right now, unless we have 10,000 people. I think somebody else had it.
Is there any other way you can compare your experimental results? Because this MMSC score is subjective evaluation by many. They ask some questions, they come out with this score. But what your experimental is highly, sophisticated. You extract results using MEG and other things. You have any other parameter to compare your results?
So for cognitive decline, this is what we have. But the other studies, all of this that I showed you is purely based on MEG and model fitting. So even like these show that there is a difference between Alzheimer's disease and controls, and the tau G parameter is the one which is significantly different. So there's less variability here. Okay. All right. All I want to tell you from here is that the increase in there's a relationship between impairment in long-range excitatory neurons and global cognitive decline in patients with Alzheimer's disease. So this is something we can try and infer using real neuroimaging data and applying mathematical model. So is there anything else we can do with the same data? So we can also do something else. By the way, this paper is published if you want to look at. So we can also look at some much faster time scale spontaneous activity. And the idea here is that you have the time series from MEG. Now instead of taking all the time series and taking a Fourier transform of that, do a time frequency decomposition. And then what you get from here is dynamics of how the power spectral density changes across time. And then we can look at a much finer time scale how the frequency spectra is changing and subsequently how the model parameters are changing, and then we can try and see use this to see if there is any difference in the dynamics between Alzheimer's disease and controls. So that's the idea. Given time frequency decomposition, same MEG data, extract frequency spectra at different time points, do model fitting, see what the model parameters are, and get a dynamic profile for all the subjects. So x-axis here is time in seconds. Every row here corresponds to a subject, either healthy or Alzheimer's disease, and the color tells you the intensity of the parameter, the value of that parameter. So basically, now by doing this dynamic model fitting, you get a dynamic profile of how the model parameters look like. So to recap, what did we do previously? We had this spectra. We did this model fitting and did some associations and found this. Now what I'm gonna show you next is we have time frequency maps, and now we are going to fit the model to every frequency spectra at different time points, and we'll get dynamic profile of these parameters, and then we'll compare them, and that will also tell us something. So, but how do we quantify these dynamic profiles? There were two metrics we used for calculating this. We looked at the sample entropy and coefficient of variation of the dynamic parameters, and we compared that between Alzheimer's disease and healthy controls. So if you see, again there are some for which it's quite different. So if you look at sample entropy of this coupling parameter, which is alpha, it's lower in Alzheimer's disease, and even the sample entropy of tau G is lower in Alzheimer's disease. Again, coefficient of variation and tau G. Now we can ask the same question. can we use these as markers of disease? Can we try and predict, classify Alzheimer's disease from controls using these as features of a classifier? So this is how the classification looks like. So in red is using the static parameters as before, and this is the area under the curve. In blue is using sample entropy of the dynamic parameters, and that's the area under the curve. In yellow is using coefficient of variation of the dynamic parameters, and so that's the area under the curve. And in green is the, if you use all of these as features of a classifier, this is how the area under the curve looks like. So it increases, the area under the curve is much better if you include all of these features as classifiers of a classifier. And, sorry? Yeah, not necessary. So we just looked at the importance to see now if we use all of them, which are the most important ones to achieve that number. So what we see is the most important features for classification really are tau g as before, but now the most important one is actually the coefficient of variation of the coupling. So that seems to be the most important one for accurately classifying Alzheimer's disease from controls, which was not a feature we used before. Now, so overall, again, that tells us that both dynamic and static features contribute, and they both contribute in classifying Alzheimer's disease from controls. And the most important contributing features are impairment in long range excitatory time constant corresponding to tau g, and the coefficient of variation of coupling, which is this alpha parameter here. So we ask the same question, can we again use these as, do these also have any clinical significance, and can they predict global cognitive decline in patients with Alzheimer's disease? Again, with the same caveat that these points are scattered, the coefficient of variation of this alpha parameter is again correlated with, I didn't mention the p values here, but I am only showing these because these are significant after multiple testing, is correlated with MMSE and inversely correlated to another metric of global cognitive decline, which is clinical dementia rating, but they both are inversely proportional to each other. So basically, all in all, what I have tried to show you is in the first study, if we just look at static frequency spectra between AD and controls, we do a model fitting, we will get some parameters which are different between Alzheimer's disease and controls. They can be used to classify Alzheimer's disease from controls accurately, and if we look at some relations between them, this tau g parameter was associated with global cognitive decline. In second part, instead of looking at the entire static frequency spectra, we can look at time frequency maps to try and understand at a finer time scale what are the dynamics of the model parameters, and we can look at the finer time scale if we fit a model to every spectra, how the model parameters are changing. We get a dynamic profile. From that we evaluate the sample entropy and coefficient of variation of model parameters, and then again use them as features of classifier, and we see that they can also classify Alzheimer's disease from healthy controls accurately. And then again we can do this association with global cognitive decline score and see that it is associated with a coefficient of variation of coupling. So both of these indicate that overall there is some impairment in the time scale of long range excitatory neurons, and there is some impairment in dynamics of global coupling in Alzheimer's disease. Also, I did not go much into detail, but the dynamic impairment that we see is essentially the coefficient of variation is higher in healthy controls, and it is also higher in patients who have lesser cognitive decline. So essentially there is some abnormality in coupling which is there in patients with Alzheimer's disease, but even in patients with Alzheimer's disease, that abnormality is proportional to the extent of cognitive decline in them. So both of these are some insights we got by fitting a model to this real data, and because we had some biophysically interpretable parameters, we could infer some things like we could get these inferences from this kind of a model. And these can be used as markers of disease, because they can accurately classify Alzheimer's disease from controls, and they both are associated with global cognitive decline. All right. Now, the whole idea of this kind of work is, using something like this, we can try and identify potential mechanistic links between abnormal neural oscillations and cellular correlates of impaired neuronal activity in Alzheimer's disease. So, abnormal neural oscillations is what you can see in real data, but what are the cellular correlates of that impaired activity? That is something you can't see with just recording somebody's neuroimaging data alone. That is where you need some kind of modeling, or you can only do that in basic science studies with rodent models. So, using this model helps in identifying those mechanistic links. Because this model is quite easy, you get a closed form solution directly in the frequency domain, very quick to simulate, quite easy to fit to different kinds of spectral patterns. So, you can really extend this and investigate various other diseases as well as brain states. So, I have worked on stages of sleep. You can also work on different cognitive processes, states of consciousness, disease, pick any disease, and you can try and investigate that using this kind of a modeling work. In fact, with not with this model, but similar models like these have been used quite widely to understand various different diseases and processes in the brain. Okay. So, if you didn't get anything so far, broadly what I have tried to show you is a model where, given the structural network, we can try and capture brain function. And if we have this model, if we have some real data, if we apply this model to real data, we can try and get some insights into what might be happening that can explain what we see with the data. Of course, I showed you one model today, but is this the best model? So, I am going to end this talk with some pointers for discussions. No, it is not. There are various models available, and it really depends on your use case, what you are trying to capture, at what scale, and what is most important for the application that you are working with. All the models are based on assumptions. So, one assumption we made here was that all the parameters were global. Now, it really depends again on your application, because what we wanted to fit to, having this kind of assumption was sufficient, and even with this assumption we were able to quite accurately fit not just to the spectral patterns but some spatial patterns as well. This assumption was fine for this study, but if you want to do something more fine grained, we have to introduce heterogeneity into the model parameters. There are various ways of doing this. For example, you can introduce more maps. There are certain maps available that tell you the extent of myelin content in different brain regions, or how neurotransmitters are concentrated in different brain regions. So, you can incorporate that to incorporate some sort of model heterogeneity, parameter heterogeneity in your model. Assumption of linearity. So, the model that I showed you today was linear, but does that mean the brain is linear? Definitely no. But all it means is that a linear model can capture the data that was recorded from the brain accurately, the data, the spectral data accurately. But it is an open question as to what processes are non-linear and to what extent, and how this non-linearity varies. For example, is the time series that you record from neuroimaging, is it more non-linear when I am resting versus when I am sleeping? So, which model is more appropriate in which state? These are all open questions, open for anybody to work on, and we all are seeking answers to these questions. With these pointers for discussions, I would like to end. So, most of the work today that I presented is what I did as a postdoc at UCSF, and this is my contact info, and happy to answer any questions you might have.
So I don't see any color bars in this frequency versus time plot or in the distance matrices.
Color bar in?
So can you please open the frequency versus time? Yeah. Yes. So this frequency versus time graph,
Okay.
or those structural connectivity matrix.
Okay. Yeah, I didn't show you, but red is higher.
Yeah. So means what is the range of the data? So you can understand.
So its spectral power, it's in dB scale. It ranges from -10 to -80, something like that. dB in dB.
Okay.
So as expected, it's much more in this 10 frequency hertz range, and that's expected given that this is for somebody who's resting with eyes closed.
Okay.
Sorry. Okay, I can ask before the mic is passed. Because okay, thanks for the presentation. It was very nice to see the frequency domain approach than the state space that we do. Okay, relating to our work, so how do you infer functional patterns here, especially in the case of Alzheimer's? And are there some kind of regions that you specifically look at from the brain?
So this was a global study, so we didn't pinpoint any region, but we have done a local study where we do focus on some regions more, and that depends on the amyloid and tau deposition in the brain and how it spreads as Alzheimer's disease progresses. So for example, tau starts in the medial region while amyloid is more diffuse. So is there any relation between regional distribution of these parameters with how the amyloid and tau proteins look like in the brain, the spatial patterns? So
because the dxk by dt, the xk was a region, right? So what is the interpretation of region?
That was a different study where we actually didn't. So let me go to this. When we did the regional study, we didn't incorporate this. We only looked at the mesoscopic level parameters. We inferred that for every region separately, and then we saw if they are associated with the extent of amyloid and tau deposition in the brain. So we can also, other than that, at a global level, compute like imaginary coherence and those network-based matrices and compare them. In a separate work, we showed that this model can also capture imaginary coherence accurately.
Okay, we can continue to this.
Yeah, please go ahead.
So the structural change, as we go from a healthy individual to someone with Alzheimer's disease, that was characterized by changes in these parameters while matching with the experimental data. Can we take another route where we are using the experimental MRI scans from an Alzheimer's disease patient, we can get these structural connectivity and adjacency matrices?
Yes. So it's a great point,
possible?
And that brings me to one point in Alzheimer's disease and what the structural connectivity is. So there are two kinds of, broadly we divide the brain into two matters, grey matter and white matter. Grey matter is primarily corresponds to function and white matter corresponds to structure. So most of the degeneration that happens in Alzheimer's disease is in the grey matter. So it's still unclear if there are network level white matter abnormal changes in Alzheimer's disease. So this structure corresponds to the white matter. So even if you get that from the data, it's going to look similar for Alzheimer's disease versus controls. But grey matter is where it would correspond to these parameter changes.
Thanks.
Hi Parul. Hi. Sorry. Just from a high level perspective, I don't understand a lot about Alzheimer's disease. What do you need to actually predict from your model, for example, going forward? Let's push it forward. What do you need to accurately predict a patient who will get Alzheimer's and who will not get Alzheimer's?
What I need is longitudinal data of progression.
Can you explain what longitudinal data means?
Oh, yes. So if we get brain scans of healthy individuals and track them over the years, how their cognitive abilities are declining, and then out of them how many develop Alzheimer's disease and how many don't. With that we can actually create a predictive model. So that is what I need. Yes. It's being collected. So there's this CBR in India, Center for Brain Research. They are collecting this longitudinal data.
Hi, Parul.
Yes, ma'am.
Go ahead.
Yes, ma'am. Thank you for your presentation. So, studies have shown that most of the neurodegenerative and degenerative processes occur in the subanatomical structure of the brain. So, thus, your model, has the ability to capture subanatomical structure of the brain.
What do you mean by soft anatomical structure?
For example, the phonex is a subanatomic.
Oh,
yes.
Subanatomical.
Yeah.
So, the MEG data that's captured is only for the cortical level. It cannot capture the inside part of the brain. So, that's limitation with MEG because it's non-invasive. So, as of now, we cannot capture that. With fMRI, we can, because fMRI gives you a much better spatial resolution. But then fMRI times, if you look at the temporal resolution, it's quite poor. Because we were interested more in something that had a better temporal resolution, we worked with MEG in this case. But you can, if you're more interested in looking at the subcortex, subcortical regions, fMRI would be more appropriate.
Thank you.
Parul, here. So very nice talk. The scanning that you mentioned, right? So what is the resolution? I mean, is it at a neuronal level or it's a much higher?
It's a much higher level.
Much higher level.
Yes.
So is there a technology that's coming up where you could probably look at neuronal level signal transduction, or that's? I mean, what is limiting there? Because we do see kind of, we can image single neurons. Typically in a petri dish, right? So,
yes.
So in terms of MRI and MEG you mentioned, right?
Yes.
So what is limiting the resolutions of those technologies to go to neuronal level? Because then you can have. Is it because even if you have the data, you won't be able to handle it? Is it because of the reason that you have too many connections?
No, it's not that. It's just I don't think we have the technology to. See, all of these don't really capture neuron activity directly. They're all capturing an indirect measure. MEG is capturing the magnetic activity of the brain, and there are only some limited sensors on an MEG machine. So, depending on how many sensors are placed, it's only going to pick up signals from those many sensors. You can, if you want something much fine-grained, you have to place something on the surface of the brain, but then you have to do a surgery. So, that's in a non-invasive manner. This
is the best we could.
As of now, from what I know.
Okay, thank you.
So a couple of questions. One is a follow-up on Nitin's question, which I'll ask later. But before that, one of the criticisms in medical world has been that a whole lot of data, a whole lot of analysis is based on male Caucasians. Does this type of work also suffer from the same critique?
So, from what I remember, gender, it was almost equally distributed. But Caucasian, yes, that the racial distribution was not
So
equal.
do you expect the results to be slightly different if that is
Yeah, we would expect. I mean, it's a criticism, it's a valid criticism of medical domain, but it's also really hard to collect this data. So we have to wait longer to get enough numbers.
The follow-up question on Nitin's question was so in for example, autism. There are a lot of studies where, for example, because autism is also might have a genetic component. So if, for example, an older sibling is autistic, studies on the younger sibling starts right from birth and so on. Are there studies like that also happening in Alzheimer's on related neurodegenerative diseases, or?
I'm not aware. It might be definitely not something from birth, but
no,
as
I mean
people
in
get
Alzheimer's
older,
it will be much older, maybe 60 plus.
Yeah.
But if, for example, the older sibling has Alzheimer's, would people recruit the younger sibling in time transient studies over multiple years and so on, and would that help?
that would help. I am not sure if it's being targeted like that, but what I know, CBR, they are just collecting genetic information, blood test as well as scans, and it's a longitudinal data. So hopefully after a few years we'll have that information, at least for the Indian population.
Very interesting. So you adjusted the parameter to match the spectra, right?
Yes.
Can you make it multi-objective? In addition to the spectra, can you also match that score, that neuron activation score, whatever the score you have? Can you optimize both of them together?
So actually, we do a multi-objective one, optimization. We not just fit to the shape of the spectra, but also the spatial patterns. So in that sense, we try to maximize two cost functions simultaneously.
No, I'm asking about in the later part when you're correlating your alpha to the score. What is that score? I forgot.
Cognitive decline.
And that's where the lot of dispersions coming.
Yeah.
So can you fine tune your model parameter to match that score also in addition to the spectra? Because that's probably a determining factor whether somebody is prone to Alzheimer or not. If that's the first order variations, why not optimizing your model on that particular properties?
That particular property is not a biophysical property, so it won't come anywhere in the model.
I see.
So as of now, I cannot, unless I figure out a way of quantifying cognitive decline in a biophysical manner.
Okay. And my second question is that I understand that you have very small amount of data, about 80, but what is the limits of that score in general, humans or any animal? Any extreme limits theoretically or experimentally identified?
So those are the limits, the ones I showed you.
About 5 to 20 or something, 30.
Yeah.
That's the extreme possibility.
Yeah. Generally, these are the limits. For healthy, they'll all hover around 20 to 30, 25 to 30.
I see.
For Alzheimer's disease, they are more varied.
Okay. Thank you.
So just wanted to understand. So since it's a linear model, like maybe a very primitive understanding is that what you put in is what you get, right? For the most part. So is it somehow saying that all the information is somehow encoded in the structure, but retrieved through some in dynamical form or something like that? And it's somehow difficult to extract these inferences just from your adjacency matrix and
it's difficult to extract just from the matrix because that is like an input to the model, right? So if you look at the equation, they govern spatial heterogeneity, but finally the dynamics are governed by these extra equations, and those parameters really control where the spectral peak will occur. It's not the structure that controls that. The structure will control the power of relative power of regions.
Yeah, so one question which Tarak was asking, so the cost function that you're optimizing, is it convex? Is there only one possibility of the optimal values of the parameters that you have, or is it a non-convex function?
The cost function is Pearson's correlation, so it's I think not convex. If it was MSE, we could have said that. We still use Pearson's correlation because we really want to fit to the shape more than the amplitude. So that's the.
So in your spectral data thing with respect to frequency, so you have analyzed about 80 patients or so. From the control to those who got Alzheimer, can you able to say like if these are the people who are controlled can have a potential to get into Alzheimer at some point of time?
In this case, these are two different populations, the same age range, some who are healthy and some who are Alzheimer's. It's not that they were healthy and then they converted into Alzheimer's.
No, based on that also, assuming that at some point this will have this kind of a data,
yes.
Is it possible to say this person has more prone to get into Alzheimer at a later stage or?
So Yeah, we can get some insight like that. So basically, Alzheimer's disease, this 88 cohort, it consists of both who were mildly impaired as well as who had full-blown Alzheimer's disease. So you can divide the population into healthy, mild, and full-blown Alzheimer's. And then you can test if there is any progression in the parameters. So interestingly, we saw that tau G has a progression. It gradually increases from healthy to mild cognitive impairment to full Alzheimer's disease.
So also in your spectra, there is at some point there is a overlap between the control and the other. If you go to that spectra, at some frequency there is a overlap. Will that tells anything? Yeah.
What do you mean by overlap? In the sense here? So I mean all of these were the frequency Fourier transform that was done. That was for these frequency ranges, right? So we'll get some line which looks like this. So at some point they might or might not overlap, but I'm not sure if we can infer much from that.
Okay. Yeah. Thank you.
So do we have any further questions? If not, let's thank the speaker for giving us a very interesting talk. And thank you everyone for your very active participation and your attention. And we'll get together again in 2 weeks of time when we'll have another speaker as a part of the colloquium. Thank you so much.
Thanks. Very thirsty.